Is Mary Frances Mahalovich. She's a geneticist for 4 different Forest Service regions. I don't know how she does it. She has quite a lot of energy. We're crowning her right now. There we go. Mary Frances.
Thank you, Liz. I'd like to acknowledge my co-authors that either provided data or prepared tissue for our analyses, or Dr. Kimsey that assisted in some of the spatial analyses that I will present this afternoon. 18 years ago, when I was asked to identify, harness, and deploy blister rust resistance in whitebark pine, our primary trait was developing rust resistance. And then 11 years ago, because of concerns with climate change, we started to assess cold hardiness. And 6 years ago, we began to also address drought tolerance because we wanted to fill in some missing information gaps to characterize the adaptive capacity of whitebark pine to better inform management decisions, assist us in our vulnerability assessments, and allow me to go back and take a hard look at the genetics program and see if we needed to make any mid-course corrections. So in the interest of time, I'll only talk about the blister rust resistance and drought tolerance this afternoon. One of the key pieces of missing information to assess blister rust resistance in our program, and to also help explain the spatial and temporal variation that I'm seeing in rust resistance, was we needed a more current measure of rust infection in the inland west. And based on the hundreds of people that have been helping designate well over 1,300 plus trees, I've also asked you folks to get data on blister rust and mountain pine beetle incidents in a 100-tree survey. So it's those 100-tree surveys that gave us the data from 343 unique plots. And the main reason that I wanted to show you this slide today is to continue to build on the momentum that even in the recent outbreak of mountain pine beetle, blister rust continued to increase on the landscape. And if we look at the results and a mapping exercise by a postdoc of Bob Keene's, Helmbrecht, you can see from 2006 to 2014, we've had a huge and dramatic increase in blister rust infection, and we hardly have any areas left with low blister rust pressure in our ecosystems. And this also helps explain the patterns spatially and temporally that I'm seeing in blister rust resistance. So here is one of the first graphics to establish baseline data for rust resistance with very low spore loads. It's data from our 110 seed source study at Coeur d'Alene from 1999 to 2005. We look at 7 different rust resistance traits, and 6 of those traits actually require the seedling to become infected with blister rust before we can assess resistance. So we're not developing the completely rust-free clean tree in our program. In reviewing other programs such as Michael's and Richard's and work that's being done in Placerville, the metric that seems to resonate with audiences is percent resistance, so that's what I've elected to model and present today. But please recognize that each of our programs defines percent resistance differently. And in addition to the stem symptoms in our program, I include the needle resistance as well as the fascicle bundle, the short shoot. So by definition, our rust resistance can be higher with the same plant material. One of the other things that I wanted to indicate with this spatial map of blister rust resistance at low infection levels is looking at seed source differentiation using a least significant difference approach. You can see that whitebark has the generalist adaptive strategy, where most of the variation in rust resistance is within seed sources rather than among seed sources. And so we have very large areas of the same color. And interestingly enough, the areas where we have the highest levels of rust resistance are also the same areas where we have the highest incidence of blister rust infection. As we go through each of these adaptive traits, as well as the molecular data for genetic diversity, it's a different suite of climatic variables that are explaining the majority of the variation. So that was the other reason for going through this exercise, to see what are the key climatic variables that are explaining the majority of the variation. It's not always mean annual temperature or mean annual precipitation. And how does that compare to the climate modeler's work that's underway in Whitebark pine? So the next trait that I want to fast forward to is drought tolerance. And the way that we assess drought tolerance in our program is through stable isotope analysis and using Farquhar's linear approximation of intrinsic water use efficiency. But you can't just stop there and say that carbon stable isotope discrimination equates to drought tolerance. You need to follow it up with field studies on stomatal control and leaf area, stomatal conductance, and we had the advantage of being able to corroborate that relationship with work that was done by Anna Sala and others and her graduate students. So we moved forward, and the first spatial representation at a very coarse level is among the seed zones. And the Greater Yellowstone area, or the Greater Yellowstone Grand Teton seed zone, is one of the most drought tolerant, and significantly so among the inland west seed zones. The other interesting thing, I'm not going to present the data on the nitrogen stable isotope or sulfur because that was more the aspect of a wildlife study, is we begin to see an emerging trend in soil parent material having a very strong signal in explaining the patterns of spatial variation in our traits. So here's what that model looks like. And again, the areas in dark blue are areas that are indicative of high drought tolerance in the inland west. And when we compared our data and adjusted, we used nut tissue because of the interest in the data for the grizzly bear researchers. So we had to apply a correction factor because most of the intrinsic water use efficiency studies are done on tree needles rather than nut tissue. Having made that adjustment, we find that when we compare our results to those of John Marshall and Jianwei Zhang, whitebark pine is one of the most drought-tolerant conifers in western North America. If it has a potential competitor for that top slot, it will probably be limber pine. I'm not going to talk too much in detail about cold hardiness or genetic diversity, and the way, given the individual tree data that we had for the molecular genetic study, the way we express genetic diversity is the percent of polymorphic loci based on 16 loci evaluated among 160 individual tree samples. So I wanted to begin to look at different modeling techniques to answer questions about an integrated representation of adaptive capacity for whitebark pine, and also to see are we doing a good job of sampling the adaptive capacity and genetic diversity on the landscape in our program. So we took each of the layers, rust resistance, drought tolerance, cold hardiness, and genetic diversity, and integrated those. Right now they have equal weightings, but in the sensitivity analysis I can tell you that some of the variables have a much harder or stronger pull on the integrated results. So there's a lot of room for improvement, but this is just the first cut at how we can use some of this data coming out of the genetics program. And the areas in blue on the right-hand side are those areas that have a higher likelihood of being blister rust resistant, drought tolerant, cold hardy, and genetically diverse. We then overlaid that map with the 1,300 plus trees in the program to get an idea of how well we've sampled the genetics of whitebark in the interior. And you can see that there's some areas for improvement where we have no plant material represented in the program. So our later rust screenings, beginning in 2008 with the Greater Yellowstone and the Central Montana seed zones, I began to group the selections by seed zones to facilitate seed orchard selections and also to prepare sheets that would indicate the rankings of each of the seed sources. And until our seed orchards reach reproductive maturity, what I do is highlight those seed sources that are in the upper 25th percentile for rust resistance. And I ask the forests and other agencies to emphasize those areas in the cone collections so that we have a higher probability of deploying rust-resistant seedlings on the landscape. But as Bob alluded to earlier this morning, if we confine all of our operational cone collections to these 7 areas in the crown over the next 10 years, aren't we perhaps reducing genetic diversity in whitebark pine? So we can use maps like this to better inform decisions of where we can bring new material into the program. We have 10-year seed procurement plans in the Forest Service, and to manage the genetic diversity, we not only should be looking at cone collection areas that spatially vary across the landscape, but also temporally vary across the landscape. So in our next 10-year planning window, perhaps we can bring in material from some of these other areas so that we're maintaining genetic diversity over time. In all of these analyses, and I don't have time to get into the methods and the results of each of the adaptive traits, I'm picking up an exceptionally strong signal for soil parent material explaining the majority of the variation in each of these adaptive traits, and not only in whitebark, but in all of our other conifers, and they also have me doing gene ecology work in native plants. And to the geneticists' credit that have done gene ecological modeling in the past, soil parent material data wasn't available. And so most of our species are characterized as having clinal variation. But having gone back and retrospectively reanalyzed all of our data, we also find that our conifer species have ecotypic variation for the various adaptive traits. So that got me thinking about some statements in the range-wide strategy, that whitebark is thought to be limited at higher elevations based on temperature, and the expansion of whitebark at lower elevations is limited by competition. Given that we have such a strong edaphic signal, is it possible that our current distribution as well as our future distribution of whitebark also modulated by edaphic variation? And if we go back to the sylvics of each of our 5-needle pines in this area, whitebark grows on inceptisols, and limber on antisols, and western white pine on ash caps or andisols. And so this may have far-reaching implications for our assisted migration, naturally or a facilitated assisted migration on where we match species to site. And perhaps some of the recent work in assisted migration with whitebark, their failures could perhaps be, even though they had suitable future climate space, they didn't necessarily have a suitable substrate. So I've also looked at the correlation among traits, and again, I pick up a strong signal for where the calceds are concentrated in the inland west for cold hardiness and drought tolerance, and where the cal metaseds are concentrated in the more northern aspects of the region. So from a geologic perspective, I further got to think, could we use this data to potentially define climate refugia? And so we know from the literature and from the paleobotanical record that limestone soils, ultramafic and podosols, are those unusual areas where species retreat in the face of extreme climate change. So Mark and I applied an additional filter to our integrated analysis. And when we add soil parent material, the areas in green are possible areas for future climate refugia for whitebark pine. And again, the black dots represent where we have plant materials represented in the program. So this might inform where we prioritize our strategies for restoration. We don't have adaptive capacity in any of our species distribution models, and I would encourage our climate models to also apply a filter of suitable substrate, and then we'll get better predictions of where whitebark will persist into the future. Another filter that we could apply in this process is density. We talked about low and high density and prioritizing areas for conservation, but I'm very concerned about if we drop below too few trees per acre, 10, that will wash out any advantages that we have in our populations and build up inbreeding. If you don't have a large number of trees per acre, you don't get an effective pollen cloud and seed set. And eventually that leads to the decline of natural regeneration and potentially local population extinction. So some of the implications of the work that we've been doing, whitebark continues with each of these adaptive characters to exhibit a generalist adaptive strategy. We need to consider not only the clinal variation in our modeling, but also the ecotypic or edaphic variation to better inform management decisions and climate models. And I think with that, in the interest of time, I'll wrap up.
Mary, this is Diana Tom back here. I know reading Arnaud and Hoff, they do mention adaptive variation across the range of whitebark pine, and that there is actually a water interaction with some soil depending on drought and local climate conditions, whether or not whitebark can successfully grow, say, on limestone, or the relationship with siliceous materials. So that probably bears further exploration because toward the northern end of its range, it is in fact excluded from some areas because of soil characteristics, soil types. So I think you're on an interesting track there.
I don't know if we've explored it fully in the drought tolerance, but one of the climatic variables that it was explaining the variation was Hargrave's climatic moisture deficient, which is the available soil and moisture in addition to precipitation. So maybe that's beginning to get at your valid point. Oh, go easy on me.
No. So I think the whole drought tolerance thing is really fascinating work because it really hasn't been quantified in whitebark pine. Ted Weaver measured in I think it was 70 to 78 water stress in whitebark pine in the Yellowstone area and found they never went into water stress at all. Yet we were sampling whitebark pine stands that were in 10 inches of precip a year in a sagebrush grassland where they were in water stress for most of the growing season. So I think its wide range of drought tolerance is one of its abilities to withstand climate change. Do you agree?
I would agree, and there's two distinct climates in the Yellowstone ecosystem, and we didn't have royal blue blanketing the whole Yellowstone ecosystem. There were areas of red that probably are those that you have the standing water and maybe aren't picking up those drought-tolerant individuals. So that's an interesting point. I didn't look at Weaver's study, but I will.
Well,
he never published.